There are many levels at which biologists can study living organisms, and there are many techniques that they can use to study those organisms. One of the ways that biologists study living systems is by the use of model organisms. So a model organism is an organism that has been and is being extensively studied. And there are a variety of reasons that we would use these specific organisms, but it's usually related to the information you're trying to gain. So you would choose a model organism based on being similar to the system that you're trying to learn about. So for example, instead of starting with studying humans, you might start with studying something like the mouse, or a nematode, which is a flatworm, or even a zebrafish. These are organisms that are common model organisms that help us understand more complex systems. There are model organisms from all of our domains, all of our kingdoms, that are going to help us learn a little bit more about living systems. So biologists very heavily rely on the use of model organisms to understand more complex organisms, or even similarly complex organisms that might be less common or less readily available. And we can use model organisms because of evolutionary history, because all living organisms at different levels can be traced to common ancestors. And so any organism that is in the same, say, genus or the same class, phylum, or order, any of these other categories can help us learn about the other ones in those categories because of common characteristics and common ancestors. And so what this means is that if we can learn about something in one organism, we can apply it to another. And so researchers have focused on a few specific organisms, and that allows us to sort of collect a huge amount of data so that we can use systems biology, so that we can use reductionism to help us understand these different features of different organisms. So why do we use them? Why don't we just go directly to the animal or the plant or the bacteria that we want? Well, the main reason for some of these organisms is that they were studied historically. So they were used because they were readily available, easy to acquire, anybody could find them, and the research just sort of accumulated on them, and we got a really good understanding of these organisms. But we can apply a knowledge between organisms because of that evolutionary history, because similar organisms behave in a similar way, similar processes are similar in related organisms. So for example, the heart of a mouse is going to function very similarly to a heart of a human. Now we might have a bird heart that in some aspects is going to function similarly because we're all animals, but there are differences. And so we can look at, say, a mouse and learn the basic function of an organ, like a heart, and then use that information to learn more easily about other organisms. So we can start in one, learn as much as we can, and then move on to something that we might not have as great of access to or might be harder to study. So because of that common ancestry, because of those evolutionary relationships, we can apply those similar processes to related organisms and to many different organisms. We often use model organisms because they are more ethical to study. Now, right now, we're not going to get into an ethics debate about whether or not we should. But from a biology standpoint, you have to study something. And so most people would agree that if you're going to study something, it probably shouldn't be humans. People have an emotional attachment to more complex organisms. And so you don't want people doing research on really cute beagles or chimpanzees. But sometimes we need to do that to avoid doing things to humans or other animals that we don't want to. And so we rely on model organisms to first get that basic understanding. And then maybe we can move on as needed and as appropriate to more complex organisms. So if you want to learn about something in a human, you might want to start with a mouse or study humans in an ethical fashion. There's that too. But the reason that we can use model organisms is because DNA is a universal genetic code. Now, what that means is, with very few exceptions, the structure of DNA, the function of DNA, and the expression of DNA is essentially the same in all living organisms. It is not identical. There are different ways or different key features that are different in the expression of genetic material, say between a bacteria and a human. However, the overall function is so similar that we can take a gene from, say, a pig and insert it into a bacteria, and it will produce the outcome or the proteins the way that we want it. And so because this genetic code is universal, because gene function is the same or very similar in closely related species, and because our structure and mechanism of expression is the same in all living organisms, we can apply information from one to the other, because we can assume that a gene that is similar in structure and function in one organism is going to do the same or a very similar job in another. That's why we can use a mouse to study humans. That's why we can look at mouse genes and determine what they probably do in humans. It's also cheaper to study animals and plants, especially smaller plants and animals. So from a practical sense, model organisms save us a lot of time and energy. If we have a basic understanding of mammals from a mouse, we can apply that information. So we don't have to go study dogs to the extent that we studied that mouse. We don't have to study cats to the extent that we studied that mouse, because our understanding from those mice gives us all of this information about these other organisms. But from an even more practical standpoint, there are a few characteristics that make model organisms really great for studying. Now, I said that many of these started out as organisms that were easily accessed. For example, E. coli is found in poop. So it's easy to get E. coli. You can take it out of poop samples. Fruit flies will magically appear on any rotting fruit that you leave out. Mice are all over the place, especially in old, dirty cities that some of these scientists were working on. And so early organisms were selected because they were readily available. They were used continuously because they contained some of these characteristics that make them great to study. One is that they are easy to grow, house, and care for. This is usually because they are small, so you can keep a lot of them around. They have relatively short life cycles, so it doesn't take a long time to study them from conception to death. They usually produce many offspring. So from a single fruit fly whose life cycle lasts a few days, you might get hundreds of baby fruit flies. And so this allows you to collect a lot of data and a lot of statistically significant data because you have a huge sample size. Straightforward genetic analysis. So what this means is really that their genetics is very well understood. The more complicated your animal is, the more genes they might have. Not necessarily, but usually we look at the simpler organisms as having more simple genetics. So for example, an E. coli has much less DNA than, say, a fruit fly or a mouse. But what we really mean about straightforward genetic analysis is that it's well understood or well studied. This might mean that the genomes have been sequenced, that many genes have been identified, their functions determined, and maybe they've been mapped within the genome. And so really we mean that they have been extensively genetically studied. We have a lot of information about them. Sometimes they have less complex systems. For example, the nematode, that flatworm on the very first slide. The nematode is really cool because its development of its nervous system has been documented for every single step in its development, which helps us to understand the more complex nervous system of our higher animals, like our mammals and our reptiles and things like that. A mouse is not necessarily less complex than a human, but it is smaller, and so that makes it a little bit easier. So straightforward genetic analysis and less complex systems is not necessarily a requirement, but it does help with making a model organism useful for studies. This is not at all a complete list of model organisms, but a few that you might have heard of. Drosophila melanogaster, that's our fruit fly. You can grow these in your kitchen if you really want to. Drosophila was really important in early genetic studies. It was also been important in a lot of understanding of molecular processes and gene expression. E. coli has been used for a ton of different things. E. coli is a prokaryote, it's a bacteria, and it's been used for all kinds of things, but most notably for us, there's been a lot of molecular studies as far as gene expression and understanding how we take the information from DNA and get it out. The mouse is a mammal, which makes it really important for human studies, especially as it relates to diseases and heredity and things like that. Saccharomyces cerevisiae, that's baker's yeast. This is a eukaryote. So not as complex as a mammal. It's a single-celled eukaryote, but that makes it really easy to work with. There is a model organism for our plants as well. That's Arabidopsis thaliana. It's a very tiny little plant. It's actually a mustard plant, and it has been used a lot, especially in genetic studies and plant development. So there's a few others. We saw the nematode and the zebrafish on the first page. The organism that you would choose really just depends on the system that you want. They're all going to have positives and negatives, but all of them are going to be able to help us have a better understanding of the other organisms that are related to it.